I've noticed a number of people have been looking at these labs here, these lab notebooks. These are here to just kind of give you a sense of what things ultimately end up looking like. You know, kind of a mix of pasting in the methods, writing in your data, you know, making graphs, if you're analyzing your data. You'll be doing this for each of the different labs as we go through the semester. Again, table of contents where you will be, you know, as we build up the lab notebook, you'll say, this is the lab we're doing, this is the page it's starting on. Does anybody not have a lab notebook in class today? You know, if for whatever reason, like someone doesn't have a lab notebook, and one of the days when we're doing an in-person lab, you just write your data on like binder paper. And you still have to record your data as we're doing the lab. But you know, there's plenty of room to just paste stuff in. Let me find. Like here, this person did not have a lab notebook yet. They were still waiting to see if they were going to get added in or not. So they recorded their data as they were seeing it, but on this binder paper, and then they pasted it in here for, you know, later analysis. So the point is, you always want to put your data as you're seeing it. You don't want to like record it someplace and then let's put it in here, make it pretty later on.
So you want to, like, in the chemistry review lab, there's part one and it says buffer systems and it explains a little bit, and then there's the experimental procedures. You want both of those pasted in, or just the experimental procedures?
So you should, I would recommend putting it all in there because it's allowed and it's useful background information, and you know, your lab exam is open notebook, so there's no reason not to have that extra information in there. Yeah, so I would put that extra stuff in there. Now in probably, like, I know some people have already written stuff up in different ways. You know, and as you keep doing this, you'll get more kind of intuition on how to approach this. If you haven't already, I would recommend, you know, keeping at least the initial data gathering, like having the methods for the buffer things, and then having your data for what you gathered for the buffers. Having the instructions for the electrolytes, then having your data for the electrolytes. Having the instructions for the carbohydrates, then the data you've gathered for the carbohydrates. And then you can write up kind of more of a synthesis discussion of what it all means. That makes it easier to connect with what was I doing and what did I find. If you gather all your data and then put all the instructions afterwards, it is harder to follow like what exactly is going on. And again, I know at this point, some people have already kind of written things in there. So, you know, just make your best of what it is now. But as we go forward in the future, it's usually clearest if you kind of have the methods, you know, then, you know, kind of the data, you know, and kind of the discussion. So,
I just recommend that people don't pre-glue in, right? So like glue the first section and then do your data, because you never know how much room you need. And then glue in the next section.
That's probably a good idea. Especially, and this one is kind of a unique one in that there are these separate sections. Like, you know, the jumping lab, there's just one set of instructions and then you just run with your data and your analysis and stuff. So this one is a little weird in having these different sections we're all doing on the same day. Again, at the end of each of these labs, whether or not we're doing it in person or on Zoom, we'll kind of do this, talk about what did we do, what did we see, and and I'll talk about kind of the main take-home messages. And those are the kinds of things you need to make sure that you fold into your discussion. These labs have the data gathering, which we've all been doing, but then there's a discussion where you talk about like what it all means. So for part one, the buffer, here is our class data. And this is all going to be uploaded onto Canvas when I get home. So you'll have access to this. This is something that you actually do want to just print out and take into your lab write-up, because this is showing the big picture, and it will also give you the class averages so you can compare, saying, yeah, our results were very similar to what we saw in the class averages. You want to kind of show how what you did relates to the overall trends. And if we take a look, and I should just for total transparency, one group had a double drop of base, so they didn't have the value for one, so I just interpolated to make it clean. But basically, here's our data, and it looks about 7 for just the water. You start adding the acid, it gets really low. You start adding the base, it starts coming back up and gets really high. So you can see water, if you add acids and bases, changes pretty dramatically. And some people were kind of curious about this idea, like, wow, it dropped a bunch and then it barely moves, and then it barely moves, and then it jumps a whole bunch. That has to do with the math of this exponential function of the log. Every drop you're adding is a linear amount of hydrogens or hydroxyls. So like, for instance, for this H+ concentration, all I did was the inverse function of pH. I said 10 to the minus pH value. So this is just solving for H+, given the pH. And then I made a little graph. And this is hydrogen ion concentration going up drop by drop, hydrogen ion concentration going down drop by drop. So if you actually look at H+ concentration, it's very linear, drop by drop. It goes up, it goes down. But pH is a weird function with this log in there. So that's why it seems like it drops, it stays down and zooms back up. It's just because it's this exponential function. How does it compare to looking at a buffer? Anybody? Yeah.
With the buffer, there's very little change.
There's very little change. Right? And in your write-up, you want to actually kind of refer to our actual data. You can say, like, with distilled water, we had swings going all the way from 1.3 to 11.5 on average. A huge range. Whereas when we use the buffer, the swings, it stayed in between 6.9 and 7.2. We had a maximum range of 0.3, whereas we had a maximum range here of like 10. Right? So in your write-up, use the actual numbers. You can, we have the numbers, so it's a way to really bring home your message about how much more stable the pH was with the buffer versus with the distilled water. And later on in the semester, we're going to talk more about pH balance. But in your body, these different buffer systems, there's buffer systems in your cells, in your plasma, in the interstitial fluids. It's the first defense against changes in pH, the first level of pH homeostasis, making sure as you have all these metabolic processes, or you're drinking lemonade or whatever you're doing, acids are coming into your body, bases are coming into your body. making sure that the overall environment stays stable because you've got all these different buffers working. So, and I think, yeah, this shows that really nicely. Again, I will upload this onto Canvas when I get home. We can try this. You know, I'd say in your write-up, the write-up for the buffer is probably kind of on its own. It doesn't relate as dramatic, you know, the other two, parts 2 and part 3, I would say in your write-up, they're gonna be integrated. So part 2, part 3, both cases, you took some electrolyte, you put it in, or part 2, you put an electrolyte in there, you could describe it. For the most part, it looks like a salt or some kind of white powder or crystal. What happens when you add H2O to an electrolyte? Say it again.
It dissolves.
So one, it dissolves. How do we know that it dissolves? You can't see it anymore. It disappears. It goes into solution. When we looked at the carbohydrate, the glucose, and we put that in water, what happened? Did it dissolve?
Yes.
It also dissolves, right? Because we saw it disappear, go into solution. It became clear. But then things are different. When we use the buzzer, what happens when we use the electrical tester on the electrolyte in the water?
It buzzes.
It buzzes. And what are the implications of that buzz? Yeah, there's ions. So now this means that it must have also dissociated as well, because we have ions that are free to carry the electrical current. You know, what happened when we put the buzzer wires in the glucose water? No buzz. So this is important, like in your discussion, make sure you kind of compare and contrast the glucose versus the electrolytes, right? They both dissolve, they both go into solution, but then there was also something very different about them. You know, this dissociation leading to ions, leading to electrical conductance versus the sugar, which didn't.
You're saying that the glucose plus the 5 milliliters of water should have had a buzz or no buzz? No buzz. Okay, perfect.
Yeah, no buzz. And that should also kind of make sense theoretically from what we've been talking about. Then kind of the last piece was we put an electrolyte in oil. What happened when we put an electrolyte in oil? So first, well, let's start with dissolve. Did it dissolve?
No.
No, it didn't dissolve. You could still see just the crystals sitting there in the oil. It did not go into solution. So it did not dissolve. And, you know, kind of did not dissociate either. So there was no ions going into the solution. So what happened when you put the wires in the oil with the electrolyte? No buzz. So in your discussion, you're going to want to also bring that observation in, you know, again, both in terms of what you saw, but then how it relates to theoretically what's going on in there. So that's, you know, that is what the discussion should talk about your specific observations and then tie them into the concepts.
Sorry, so just to clarify, we can just do sort of like one discussion for the entire, or at least for part 2 and 3, we can do one
Yeah,
discussion.
I'd say it makes sense to do kind of a
Okay.
combined discussion for those.
Okay.
And then, okay, we only got like -5 seconds here.